Hi guys, I hope you all are doing okay. I'm going to start off with doing some notes for DNA, RNA, some transcription, some translation. So basically what this is doing is we've seen, wait just a minute. We have seen Punnett squares and we put letters there, right? If it's a dominant trait, we use a capital. If it's a lowercase trait, if it's a lowercase trait, it's recessive. So what we're looking at now is, okay, well, how do we get this capital letter or this lowercase letter? Like if you have brown hair, what's the actual code that codes for the brown hair that maybe makes that dominant? All right, so that's what we're going to look at today. So the first thing is we need to look at our DNA. And I've got some DNA strands in here, some models that I'll show the students. And it just kind of shows how it's a double helix, how there's these bonding of two pairs here. And that gets us started right with where we're at right here. Adenine will always form hydrogen bonds with thymine. We use that as a T. So oftentimes we'll just put a T there instead of writing them out. So a lot of times we'll just see, hey, A goes with T. Now this is in DNA. Okay, so we have DNA and we have RNA. And some of you may have heard some things about RNA because of the COVID vaccine. They've talked a little bit about RNA here lately. Guanine will always form hydrogen bonds with cytosine, or what is C? So in DNA, we will always say that G pairs with C. Now, you've not had chemistry yet that I know of. I know here you haven't, or if you've had some, you've not gotten too in-depth in it. So all you need to know is that there are these chemicals, we call them, adenine, guanine, they are chemicals. They can bond. Now, some of these need 2 bonds, and some of them need 3. That is why G will never pair with T or with A, and C will never bond with A or T either. That's why they bond together. It's all because of these bonds, and that's why you'll never see them bonding any other way than this. All right, so what makes up the side of a DNA ladder? Because if you look at it, you have a double helix like this. So if I had A here, I would have T right here. It's double stranded, and this is how it pairs up. This is how the 2 strands pair up, is through these letters. Now I could have A, and then this side would be T, T, T. Okay, so then what makes up the sides of the ladder? This right here, the side. So the side is made up of a sugar molecule, which is just a lot of carbon, hydrogen, and oxygen. So we have a sugar molecule there. It tends to look like a hexagon there. And then we also have a phosphate. So once again, my chemistry kids would know what this is. You guys may not, but it is 1 phosphorus with 4 oxygens, and they're all connected. So it's a PO4. Now that I'm not too concerned with, I'm just trying to let you guys know what this stuff is. All right, so why can't we use DNA to make protein? So basically, why do we need RNA? And if you've heard about the vaccine, they've done a lot of talking about how they're putting mRNA into your cells to help make these proteins. So why can't we use DNA? Why do we need RNA? And it's really as simple as DNA is too big, too large to fit through the nuclear pore. So we could say to get out of the nucleus. RNA is single-stranded. And can fit through the nuclear pore. Alright, so when we inherited cells, we saw our nucleus, and if you take biology too, we go a little bit more in depth in that, and we look at the nucleus and we can see that there are holes in it. There are these little nuclear pores. Because somehow the information, your DNA holds that information, somehow it has to get out of there. So this is how it does that. We have RNA, it's a single strand, so instead of this, it would just be that. And of course, then it's just going to hold half the letters. So RNA is single-stranded and can fit through the nuclear pore. All right. What are 3 differences then between your DNA and your RNA? One we've already said. RNA, oh, now let me make sure I write that. RNA is a single strand. Whereas DNA is double. So it's larger. Your DNA is larger. Your RNA still, though, holds, and when I show you how this happens, your RNA still holds your instructions. All right, number 2, RNA has a U. The chemical is called uracil, but it has a U instead of a T, which is that thymine. So basically, anywhere that you normally would have seen a T, you will now see a U. So I always tell kids, if they struggle with this, when we start to transcribe and translate, if they're struggling with this, anytime you have RNA and you see that there's a T, you know something's wrong. You know that can't be right. Just change that T to a U. That's more than likely all you need to do. All right, and the third thing, the reason why it is called RNA instead of DNA is because of the sugar it has. RNA has ribose sugar. It's just a little bit different form. I believe it, I'm trying to think, I'm not quite positive. I think that it's got 5 sides instead of 6, but it has a ribose sugar, whereas DNA has deoxyribose. So these letters, RNA, they stand for something. DNA stands for something. DNA stands for deoxyribonucleic acid, whereas RNA is just ribonucleic acid. All right. So if you're still writing or need a little bit more time, you can pause it because I'm going to get rid of all of this that I've written and scroll the page down. All right. And what's really cool about this is you're constantly making cells. Right now, as you're sitting here doing this, you're making cells. So your DNA is making protein all the time. So you're doing what we're getting ready to do right now. You're going through this process all the time. So we're just going to go from our original strand. So this is like DNA strand 1. And we're going to go to what is the other side of our DNA. So it's kind of like, I know that blue side. Let's do this. I'm going to put this in red. DNA 2. So now I'm trying to figure out what is that other side of my DNA. All right, so let's look at this. Now I know, I'm going to write it up here. I said that A pairs with T and G pairs with C. So let's look at this. A would be T, T would turn into A, and C would turn into G. So tag. Now right now we should not be seeing any U's at all. All right, so now I'm going to go C, G, my G turns into a C, my A turns into a T, and you can keep going. So these 3, and I've got them broken up in 3s, because this will be called a codon. All right, my codons come in sets of 3. You need 3 letters to make an amino acid, and then of course those amino acids, if you keep adding them together, they will make the protein that you need, whether it be hair, skin, whatever your protein is. G will turn into C, T, or A will turn into a T. This is going to be A, T, G. I'm going to stop doing the arrows now. You guys kind of get the drift. Hopefully you're getting the point here. C, G, G, T, 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 G, T, G, and C, T, A. Now, if I asked you guys to turn this into messenger RNA, say like, we're always making new DNA. We saw that anytime our cells go through mitosis, we have to make more DNA. So they're going through this process. But if my cell's like, oh, I need to make some protein, and I need to get this message outside of the nucleus. They're going to turn this into what is called messenger RNA. And now, A would normally go to T, but we said we can't have T. It's now a U. T is still an A. I can still have an A, and that's what gets kids sometimes when we're doing this, is they will see that and they get that mixed up. My C and my G though, it's the same as before. So really I'm gonna look at the T's here, there's A's, they still turn into A's. And if you notice, and a lot of kids do, it's like, look, your mRNA strand is the same as this first strand of DNA up here. Once again though, let's find an A here, see this A? It does not turn into a T anymore, it is turning into a U. I know kids struggle with that, and there are times where they forget and they accidentally do a T, and I'll tell them, hey, you're an RNA, you can't have T's. But that's easy to fix. Just go back, erase your T's, and put U's. All right, so we are going to draw. You guys will see my amazing artistic skills here. I'm going to erase this. Let's put that up as high as we can get it. All right, so what I'm going to draw right now is I'm going to draw a nucleus. So let me I'm going to do a large cell. Okay, so here is my cell. I'm going to draw a large nucleus here, so I'll label that nucleus. This is my cell membrane. There's my nucleus. So inside of my nucleus, I'm going to have some DNA. And now I'm going to show you. Oh man, I do not know what happened. What just happened? I'm going to redraw this nucleus because our nucleus has maybe not this large, but they have these nuclear pores where DNA cannot fit through there, but RNA can. So what is happening here, I'm going to draw my DNA. I'm going to kind of draw that wide there because I'm going to show you what's happening with RNA. So this is DNA. All right, so let's make up some letters, okay? I'm going to do 2 codons worth here. I don't know if I have enough room, but we're going to do 2 codons. So let's say that this is, I'll put it out here, A, T, G. I'm going to have to make this nucleus a little bit larger because I am going to run out of room here. So A, T, G. Let's say we then have another codon that is C, C, A. So I'm just making up 2 codons here. All right, so when I make my messenger RNA, we're going to have a line here. Of course, your cell is doing this. That A is now a U. This T becomes an A. That G becomes a C. This C is a G now. This C is a G now, and that A is a T. Now it's a little hard to read, but it is UAC GGT. So I'm going to just write here that this is messenger RNA. Okay, so I'm just kind of making sure that we do label a few things so we know what they are. So what happens is that this messenger RNA is going to go out of the cell here. And now this is the cytoplasm. Remember, I would have gold G's out here. There would be a rough ER. I'm not going to draw all that. I just want to draw and show you guys what's happening to the DNA. So the question I would ask the class is, okay, well, this RNA, messenger RNA, is going and it's wanting to send this message to make protein. Who in my cell is going to help make proteins? And if you remember back in our cell section, that would be a ribosome. So it's going to find a ribosome. So this is what I'm going to draw. Here's my messenger RNA. It's out here in the cytoplasm now. I'm going to do my ribosome in green. And so ribosomes, we learned this in biology too also, are made up of kind of 2 parts. There's a large subunit and a small subunit. We in biology 1 just draw them as little dots everywhere, and that's fine. But in biology 2, we look a little bit more in depth at this stuff. So that's what's going on here. So I'm going to label this. This is my ribosome. It's almost like we've zoomed in on that little dot. Okay, once again, this is my mRNA. Now I'm going to draw my first codon. My first codon, if you look up there, is U, A, C. All right, now I'm going to do a little star down here I want you to write. You are going to use this, your mRNA, to code for what we call amino acids. So, it might sound strange to you, but this is your body. Like, it's good to know about your body. Even if you don't like math or science, you're never going to go into a science field. It's good to know about yourself and how things are working. So you're going to use this messenger RNA strand to make amino acids, and these amino acids are just, like I said, little chemical balls that you stick together until you get proteins, such as your skin, your hair, all that kind of stuff. Okay, so what happens here with your ribosome? It has the message. This is the codon. Okay? And what it does is it sends down this little, what we call a tRNA. So let's see here. I'm going to draw it. Now this thing I usually always draw awful here when I'm doing it. Yeah, this is not a very good drawing of a tRNA. So what happens is this tRNA has what's called an anticodon. So let me label this guy. This is my tRNA. I know there's so much going on right now, but we will do plenty of practice with this. It's going to bring down this amino acid. So a lot of times you will see me write this as AA, and it's this circular blob here. Once again, it's a chemical, and you stick all these together, and it's going to make U. All right, so the anticodon is going to be A. Once again, this is RNA. It's tRNA. So A, I'm going to change to a U. I cannot have a T anytime I have RNA. AUG. And that AUG, since the UAC coded for this particular amino acid, that's what the AUG brought down that amino acid. It's really important that you know that the blue, that the mRNA is your codon, and that is what I'm going to use to figure out my amino acid. All right, now the next three letters I had here were GGU. So what happens is the ribosome moves down this line of RNA, and it continues to bring these tRNAs. So what we have is we get another one of these guys. Maybe it just read this one. And so it just brought down an amino acid, CCA. All right, and it brought another amino acid down, and you get these chains of amino acids. So amino acid make a protein chain. Now your body is really amazing when it does this because it has a start codon and it has a stop codon. So when it knows what it's like, oh, I need to make this protein, it will go to the DNA, the section of DNA that it needs, and there will be a start codon for that, and your ribosome knows that. And it will start reading your DNA, and then it will hit a stop codon. And it will stop, and then your protein's done, and then it can go outside of your cell and be used however it needs to be used. So it's really amazing. Remember this, we're talking tiny, like your cells are small. So I'm drawing this on the board, and I think it's hard for kids to visualize sometimes because it's so small. It's happening in your body everywhere, but yet you can't see it. So anyways, this is a super important part of how you work and how you thrive. All right, so there's two parts to this thing, and I want to make sure that we have these down right. So what happens here in the nucleus, this is called transcription. Transcription. I'm running out of room there. It should be ION. Transcription. What happened out here? So basically, they transcripted, it came out here, and then we translated it into our protein. So translation is what it's called out here. All right, now I wanted to draw this out and show you exactly what is happening and going on in your cells. From here on out, what will happen is I will give you maybe a DNA strand, and I will say, what is your messenger RNA? And sometimes I might ask for the tRNA, but even if I ask for the tRNA, remember, you are always using messenger RNA to get that codon. Now I'm going to show you how to get this codon. So if you look here, my codon is UAC. So I'm going to go over to a codon chart. I've got this. It's going to be posted for you guys. There's two different ways, and I posted both of them because different students like different ways. But if I'm looking for UUC, look at this, first base over here. There's U. Here's the second, and they call them base pairs, so second base, UU. So I'm in this box. My third C, it tells me that is the amino acid. Now it's phenylalanine, but you don't have to write that whole, that would be crazy if we had to write these out every time because they're pretty long words, some of them, phenylalanine. So we will just write PHE, and that is completely fine. If you want to use this chart over here, let me find UUC using this chart. Okay guys, where'd it go? Here we go. All right, so this chart now, I know a lot of students like this one, but like I said, it's just kind of your preference. You start in the middle, so here's U. My second one was a U, so you just work your way up, U, and here's C. So once again, phenylalanine. Now it will tell you the whole word on this chart. You just need that PHE. That is all you would have to write down. All right, so anyways, these are your notes. I will do an example with you when we, probably tomorrow have the worksheet. So anyways, just let me know if there's anything you need, and I will get back with you. Have a great rest of your day.